Okay, now as I was saying that this is one of the most important topics. So uh, m- when you see in your syllabus, this topic is called as the tropic responses. Basically, it's a topic of plant coordination. Now, my question is, we've been talking about the coordination much lately, and almost three of our previous lessons were on the topic of coordination. Like, have you ever seen the plant coordinating with the environment or the plant coordinating with any other stimulus?
Could it be in the sense like coordinating as in adapting to the environment?
Yes, yes, yes. The sunlight is one of the very major factor.
I've seen like
But
plants.
mhm,
I don't know how to explain, but like even the simplest thing, like the roots, like the roots
mhm.
are positioned in a certain place where it's not interrupting something that is happening before. Like if
Let
you
me
look
make it
at the
easier for
plant,
you. Let me make it easier for you. The plant coordination, the example of plant coordination that you want to talk about, I have even more generalized example. You may have seen the plants that are coiling around an object, maybe a rod, maybe a stick. Now, what is making them coiling around an object? That basically is the result of the coordination, and those are basically called as vines. If you try to know about the word, that these are called as vines. They coil around an object like the stick or a rod, and they would be climbing up the wall. You may have seen many types of vines that are climbing up the wall. Now, what is that? That is also the response of touch. That is also the response of the touch the plant is getting from the wall. Obviously, the touch the plant is getting from the wall, and it would immediately try climbing the wall. These are called as creeping vines. So obviously, these kinds of responses, these kinds of phenomena, they do exist in plants, but we don't pay attention to it. The tropic responses typically mean turning responses, turning in response to a stimuli, to a stimulus. That is called a tropic. So, would you excuse me for a second, please?
yeah. Okay.
Okay. So, turning in response to a stimulus, tropic. See, the tropic word is quite widely used in biology, and that particular word is uh, we although haven't discussed that word in our syllabus yet, and this word would is not going to be appearing in any of the further topics, but this particular word is also used for some hormones in humans. Some of the hormones are there that turn on a procedure that is not happening before, that is not happening previously. They would be turned on by some kind of hormones, and those hormones are called as tropic hormones, which means turning hormones. They are either turning on or turning off something. So let's have an idea what kind of responses these are. See, always remember that tropic responses can be positive, or negative. Uh, Positive means the object is moving or responding towards a stimulus, towards the stimulus. So that is something like, let's say you saw a thing, an animal saw the food, and he jumps towards it. That would be a positive attitude. Then negative. When you are responding away from the stimulus, that is called as the negative tropic response, negative tropism. So let's suppose that we have two types of stimuli. One stimulus is the light, and the other stimulus is the gravity. Light and gravity. Now, the light and gravity, these are the factors that I would say um, the plant shows the response to. Suppose if I talk about the two parts of the plant, one is shoot and the other one is root. The shoot is the part of the plant that is above ground. Anything above ground would be counted as shoot, and anything below ground would be counted as the root. Okay. Now, when we say that these things are basically the shoot and root, then always remember that when we talk about the shoot, I would say that the shoot would always show positive phototropic response. Positive phototropic response. What is the meaning of it? Positive means towards, photo means as from photosynthesis, light. Tropic mean turning. A response in which the plant would turn toward the light is called as positive phototropic response. Is that clear?
Yes, clear.
Similarly, if I say that shoot would show negative, basically, I missed one thing here. I needed to tell you the tropic responses are of two types. One is called as phototropism, and the other one is called as the gravitropism. The phototropism is due to light. Here, the stimulus is light, and in gravitropism, the stimulus is gravity. Now, the shoot would show negative gravitropic response. Can you explain what does it mean?
Gravitropic response,
Hmm.
something to do with gravity. It would shoot, the root would shoot upward or downward.
The shoot would show a negative gravitropic response, means that the shoot would the shoot would be going away from the gravity. In gravitropism, the stimulus is gravity. So what is the shoot doing? The shoot is moving away from the gravity. So if it is moving away from the gravity, it means it is a negative response.
Okay,
Is that clear?
yeah,
Let's suppose that you some kind of food is cooked at your home and you avoid eating it. That is called as negative tropic response, whatever kind of tropic it is, but it is negative tropic response. Since you are moving, you are uh, trying to move away from it, you are trying to avoiding it. Similarly, the shoot, the part above the ground, the part above the ground is shoot. It would always be showing the response opposite to gravity or negative gravitropism. Similarly, when we talk about the root, the root would also show two responses. It would show a kind of positive response and if it would show a kind of negative response. Now, my question is, to what stimulus the root would show positive response and to what stimulus the root would show negative response?
Positive response would be if the roots shoot downward,
Okay, then what is the stimulus there?
the gravity.
Yes, so you would say the roots would show negative, sorry, positive gravitropism as they move according to the gravity. And what kind of negative response?
If the roots shoot up,
I'm sorry.
if the roots shoot up,
The roots would show negative phototropism. They would show negative phototropism as they would move away from the light. They would move
okay,
away from the light. So now it means that I would say the root would be an roots would be examples of the negative phototropism, but an example of the positive gravitropism. That is a clear-cut thing. And I guess you can easily understand due to the movement of the due to the through the movement patterns of the root and shoot. The root would always go underground, deep underground. Why? Responding to gravity. The shoots would always go higher above the ground, responding to light. So it definitely would mean that they both are showing positive response to the stimulus they are getting. Like the light towards the light, they are showing a the shoot is showing a positive response, positive phototropism, but towards the gravities is showing the negative gravitropism. Similarly, for the roots, the root would show the negative phototropism, but they would show the positive gravitropism. Is it clear?
yeah,
Now, the point is, the point is, the most important thing here, the plants don't have specific receptors, specific receptors like an animal to turn towards gravity or light. Okay, if I say that we have the photoreceptors, how would you interpret it? See, the eyes are called as the photoreceptors. The eyes work as the light receptors in our bodies. So it means that they certainly are the photoreceptors. Light receptors, photoreceptors. Maybe we also do have pressure receptors that make us feel the gravity sometime. When we have more pressure on our joints, we feel that a lot of gravity is working on us. So the plants don't have those kinds of receptors. The plants don't have nervous system. Don't have nervous system. So how do they respond? This is the biggest question. If they don't have nervous system, then they how do they respond? The biggest question is here. Okay, let's say the plant coordination is majorly chemical coordination. meaning that the plants would perform their action with the help of the chemicals. Chemicals. They would do their work with the help of the chemicals. The chemicals would be there to show or to bend the plant. Remember, these chemicals would help to bend the plant organs. The plant organs would be bent accordingly where the hormones are located, where the hormones are present. They would be bent accordingly. Now, the real question is, how does it work? How does this work? That is the biggest question. Okay, let's say the most important hormone, the most important hormone that work in the plant is called as the most important hormone that work in the plant is called as the auxin. This hormone have a very particular function. But before knowing this function, we need to know a few things. Please be very attentive while listening to me. The effect of same hormone can change depending upon depending upon the type of receptor to which it is attaching to which it is attaching. Okay, now be very, very careful while listening to me. Suppose this is a receptor alpha, and this is the receptor beta. Both are in same body. Locations could be different. Locations could be different, but the body is same.
Okay.
So
Your voice.
the sorry, I just my voice just I guess vanished. Okay, so basically the plants, no, not the plants, the receptors matter a lot. Suppose that this is a chemical X. It goes and attaches to this receptor, and it begins a response here, begins a reaction. The same chemical X, when attaches to the beta receptor, it would stop or cease the action. Remember, the chemical is same. The chemical is same, but the response is different because the receptors to which it is attaching are different. Is it clear?
Yeah.
All right. Now, this was one concept that we needed to clear before talking about the action of the auxin hormone in the plant. The second thing is about the growth of the plant. Let's say this is the shoot. This is the shoot part of the plant. When you would observe this particular part, you would come to know about a very strange detail, and that is the plant tip. The plant tip is called as apex. And in the very initial portion, there is a continuously dividing tissue in plant. Continuously dividing tissue. This tissue is called as meristematic tissue. This tissue is called as meristematic tissue. This meristematic tissue is, like, you can say, whenever you listen the name of the meristematic tissue, you should always be thinking about a continuously dividing tissue. What is the meaning of continuously dividing tissue? It has a lot of immature cells, and they continuously divide like this. Like this. So they are dividing and dividing, and all the cells that are being divided are moved down. They are moved downward. All these cells are moved downward. Just hold on. All these cells are moved down. When they are dividing, they are moving downward. So you would say this is the phase of division. Over here, the cells are continuously dividing. Then, hold on, let me open the picture so that I don't have to draw much. Yeah, look at this. This is the this is the point of uh, meristem. This is the point of meristem where the cells are continuously dividing, and the cells, while dividing, while dividing, the cells release oxygen. The cells release oxygen. So now, be very focused here. When the cells are moved downward, they over here the cells are dividing. Now they would be moving downward. When they move downward, the cell would become enlarged. So you would say this is the zone of enlargement. Over here, the enlargement is occurring. Then the cell would move a bit downward, like even more downward. When they would move downward, the cell would start elongating. This is the zone of elongation. Elongation. You can say this is the zone of enlargement and differentiation. And the last zone is elongation. Why the elongation is required? It is the biggest question. Why the elongation is required? See, whenever you are going to try to increase the length, to increase the shoot length, we need to elongate the cells. Simply giving an example, how the humans gain height. Can you tell me that?
Cell growth, like cell regrowth.
Sorry, can you repeat again?
Cell regrowth,
Yes. Or you can also say The bones elongate. The bones elongate and the height would increase. The height would increase simply. Now, the problem is that uh, in the plants, they don't have bones, but still they have cells that would elongate and would increase the length of the plant. And this would be, now please remember, the elongation require the auxin hormone, meaning that I'm talking about shoot. Please remember, this is going to be a misinformation if I don't write shoot here. In shoot, the auxin would stimulate the elongation. What does it mean? If you go back to the picture, the auxin would be moving down here. This is the region where auxin would concentrate. Auxin would concentrate and would cause the elongation. The question is why the auxin is not causing the elongation in this region. It's a very valid question. Remember that in this whole region, cell is not fully differentiated. Cell is not fully differentiated, so the receptor does not exist. For each hormone, receptor is the first and last thing. So for auxin, the receptors have to be there. Be developed. The receptors have to be there. If the receptors are not there, the auxin would not work. So as the cell moved downward, they differentiate. They would differentiate along the way. The auxin would start working in this region, which is called as the zone of elongation. And when the cells would elongate, when the cells would receive the auxin, they would elongate. Now, this is the whole point here. This is the whole point of the discussion. That when the plant would divide itself in the tip, it would also release the auxin. But the auxin would start functioning in this particular region and it would elongate the cells, causing the length to increase. Okay, so but this phenomena would be a bit, it would become a bit other way around when it comes to the shoot. Very strangely, it it becomes working in the opposite direction. But okay, let's simplify the thing. Let's suppose, okay, first of all, save the link.
Yeah, done.
So in the root, the auxin, due to receptors, inhibits the elongation. Extremely strange, but it is true. Like I said, there was a chemical X. When it was attaching to the alpha receptor, it was increasing some sort of reaction, the rate of a reaction. But when the chemical X would attach to the beta receptor, it would inhibit the same reaction. I have a very typical example, very important example. There's a hormone adrenaline, and I told you this, but not in this perspective. Adrenaline, when it increases, when it is released in the body. Be very careful while listening to me. When the adrenaline is being released into the body, in some parts of the body, the adrenaline causes vasodilation, and in the other parts of the body, the adrenaline causes vasoconstriction. Can you tell me in what parts it would be causing the vasodilation, would leading to the vasodilation?
Um, so in the skin, the blood vessels in the skin.
In your muscles, the adrenaline would be causing vasodilation. Why is that? Because adrenaline is a hormone that would make you able to run from something or fight something. So when there is adrenaline, you would be able to fight something. So your muscles require more blood. So adrenaline would be uh, causing the vasodilation in your muscles. The same adrenaline, when goes to the stomach, it causes vasoconstriction. Now, what is the difference? The hormone is not changing, but the response is changing. Can you tell me what's the difference here?
I'm not sure.
The difference is that the receptor at which the adrenaline is attaching to, those receptors are different. The receptors in muscles are attaching the adrenaline and leading to vasodilation. But the receptors of adrenaline in stomach, when receiving the adrenaline, they are causing the vasoconstriction. That is the effect of a hormone. Is it clear?
Yeah. Okay.
Similarly, when it comes to the auxin, in shoots, the auxin would promote the elongation, but in roots, the auxin would inhibit the elongation. Have you processed all this information, or you need a minute or two?
I'll go through it.
Sorry,
I'll just go through it. Okay.
you need to repeat it. I'm sorry, I could not get your point. Your voice is a bit shady.
Oh, I'll just go over it after the class.
Yeah, okay, we'll just go over it. So let me repeat my sentence that the receptor means a lot. If the receptor is activating, the process would be activated. If the receptors are deactivating, the process would be deactivated. So in the shoot, in the stem, the auxin is attaching to the receptors, which are activating the elongation. But in the shoots, the auxins are attaching to the receptor, which are deactivating the elongation, but it does not mean the elongation would stop. That's the point. That is the point of confusion. Now try to understand the next point here. Let's say, suppose that, hold on. Yes, this is the shoot, ground, the shoot. Okay. Now, when you would magnify the shoot, when you would magnify the shoot, you would see that The light is coming from all directions. Now in the chute, the oxygen would always accumulate where opposite to the uh, point of light, opposite to the point of you can say point where the light is falling, where the light is falling. Please remember this sentence, it's very important. The light is falling at the tip of the plant. Now, oxygen would accumulate not at the tip, no. The oxygen would accumulate the point considerably below the point of light receiving. Over here, the oxygen would accumulate. This is the behavior of cells basically. If the light is falling at some point, the cells would cause the oxygen to concentrate at a different point, not at the same point where the light is falling. So you can see the light is falling at the tip, but the oxygen is being accumulated considerably below the point of light falling, the light reception. What does it mean? It means that if oxygen is accumulating at this point, let's suppose this is point A, the elongation would happen here. The elongation would be occurring here. Because what I said, the oxygen would promote the elongation in chute. The oxygen would promote the elongation in chutes, but it would never accumulate it at a point where the light is falling. So the cells would release the oxygen in such a way that oxygen would diffuse downward. You can say the oxygen would diffuse opposite to the point of light reception. It would diffuse downward. And since it would diffuse downward, the cells would elongate at a lower point, at the point lower than the point of light reception. Is it clear?
Yeah. Okay.
But it would happen totally different in the roots. Now, what I said, please remember this sentence. Oxygen is going to inhibit the elongation in the roots. Now, consider that these are the roots. This is a root. This is a root. You consider this part and you magnify it. The initial part of root is called as root tip. This is root tip. Now, if you imagine all the gravity is falling from all directions, all the gravity is basically acting at this point. Now, please focus here. In the root, the oxygen would accumulate where the gravity is highest. Gravity is highest. And highest gravity is at the tip of the root. Highest gravity. Since there is highest gravity, now the auxin would be accumulated here. The blue one is the auxin. What I told you about auxin action in the root. Would it elongate, promote the elongation, or it would inhibit the elongation?
Um, if
I'm sorry.
if needs to be present,
If you read the sentence in the black again, you would come to know that auxin would inhibit the elongation in the roots. Okay, now it means the point where there is highest gravity, the auxin would accumulate there and it would totally inhibit the elongation. Now the point is when the auxin would inhibit the elongation, the rest of the cells over here, elongation is continued without auxin. Why am I saying this? Because if there is auxin in root, it would inhibit the elongation. But if there is no auxin in root, it would its absence would promote the elongation. So you can see that when all the auxin is accumulated at the point of highest gravity, all the cells at the tip are no more able to elongate. But the cells before it, the cells above it, since they have a very little or no auxin, they would no more be receiving any auxin. They would elongate in the absence of auxin. That is the point. That is the difference between the root and shoot. In the shoot, the auxin would accumulate opposite to the point of light reception. It would promote the elongation. But in the root, the auxin would accumulate at the point of highest gravity, but it would inhibit the elongation, leading to the elongation of those cells which are not receiving any auxin. What do you think? Is it understandable?
yeah,
Okay. So basically, that is the chemistry of plant coordination. That is the chemistry of turning. Why am I saying that this is the chemistry of turning? Suppose that you have a box with a slit, and this is the over here you have a plant pot. This is a plant pot, and over here you have a shoot. The light is coming from this direction only. The rest of the portion is dark. Now, what is happening here? The the shoot would bend to the direction of light. Why? Why? See, when you would see the light falling from this direction, remember it's the shoot. The light falling from this direction, where the auxin would accumulate if the light is falling from the right side.
where the oxygen is falling.
Where the auxin would be concentrated if the light is falling from the right side.
So oxygen is transported the shady down the shady side.
Shady side, exactly. This is the side of light falling. So the oxygen would accumulate opposite to it or the shady side. So this is oxygen. Now it means since it is a shoot, it means that the elongation would only happen on the left side, not on the right side, leading to more pressure, leading to more pressure on the right side, more pressure on the right side due to cell walls, more pressure on the right side, and ultimately the shoot would bend to the right. The shoot would bend to the right. Got it?
Yes.
But talking about the root, if this is the root, suppose this is the root, and obviously the gravity right now is only from below, is only from below, and this is the root tip, this is the root tip. Now, as I said, the oxygen would accumulate at the point of highest gravity. So the oxygen is going to be accumulated or concentrated on this side, leading to inhibition of elongation. Inhibition of elongation, and elongation would happen here. Elongation would occur here, leading to more pressure from upper side, causing the bending of the root. This is why it was really important to understand that auxin would act differently in different parts because of the receptors, and it would always accumulate opposite to the shady side, I'm sorry, opposite to the light side, or it would always concentrate at the shady side, at the shady side, and and basically it is opposite in gravity. It would concentrate at the highest point of gravity, leading to inhibition of the elongation. Consequently, elongation would occur in opposite cells, and it would bend the root. So the whole chemistry of this process has been explained here. Let's go to this particular experiment. A very important one. We have understood all the chemistry of it. Chemistry, biology, everything is understood. So these experiments are literally very easy. A few things need to be explained in this experiment. One thing is what kind of um, uh, I'm sorry, what kind of equipment we are using, and secondly, what kind of groups we are using. If you see on the left side column here, one thing to consider is that we are um, using an apparatus called as clinostat. A clinostat is a drum that is movable and rotatable. You can rotate it as well as you can move it. Uh, at at the top of the clinostat, you can see this thing. It has a 360-degree movement, and this can be moved at the like the ball and socket joint of our shoulders. Now, if you focus in this particular highlighted section, you have two setups. In one, you are not rotating it. You are not rotating it. And in the other, you are rotating it. In this part, let's say it is A and this is B. The gravity is only falling on the seedlings. These are seedlings, remember, and these are roots. The gravity is only falling in A from below. Or you can say the gravity is only falling on one part of the plant, since you are not
Yes.
rotating it. Now you can see that since the gravity is only falling at one side of the plant, the root is clearly showing the positive gravitropism in A set. Is it apparent?
Yes.
In the B setup, why the roots are straightened? Can you tell
Uh,
me?
because it is rotating, so the gravity will be taking place, well, taking effect.
The gravity is equally falling on all sides of the root, meaning that it would clearly moving in the straight direction, just like the natural plant. Is it clear?
Yes.
So if I ask you, um, why did we use the mm, hold on, why did we use the B setup? We were proving the point that yes, the in setup A, the root is bending downward towards the gravity. So why did we use the B setup?
To show that um, the effect of the root when gravity is affecting all sides of the
If I ask you, if I say that um, I told you about two groups of an investigation. One group is called as experimental group, and the other group is called as the control group.
is B the control group?
Yes, because we are not changing the independent variable. We are equally dividing like we are equally distributing the independent variable to all the sides, leading to the concept of control setup, control group. Now look closely on figure 15.3 and see what's happening here. You have to look very closely. Remember, there are a few things if you miss that would be leading to wrong conclusions.
The reason uh, the second one is upright is because I'm assuming that thing is the clinostat is rotating, so that there's light
Clinostat,
on all sides.
clinostat is rotating, and
So the plant has light.
uh, meaning that light is falling equally to all the parts of the shoot.
Yeah.
So, what effect would it have on oxygen?
Mm, the
In the
oxygen,
answer to this, you would say it would be concentrated equally on all sides.
yeah, that's why it's growing because
And what are the consequences? What is the result of it? If it is distributing equally on all sides of plant, then it would go straight. It would grow straight.
yeah, it will be elongated.
There is yes, there is no bending in the plant. While in setup A, what are you looking at? No rotation of the plant.
Yeah, so there's light from only one side.
Where would the oxygen accumulate? Remember, you have to tell the distribution of oxygen in the answer.
Oxygen will include on the other side, the shady part.
Yes, the shady part, yeah. And the cells would elongate there, leading to the bending of the shoot towards the light. Okay. So this was the experiment. A few things are here. Sometimes they ask what are the improvements, like what should we consider in this experiment. The number of seeds should be equal in the 15.2. The number of seeds should be equal. The age of the seeds should be equal. A large number of seeds should be used so that if some are not viable, then others should show the result. And identical conditions are necessary. These things sometimes they ask for the improvements and consideration, so you must keep these things in mind. Okay, now okay, leading to this part. Okay, now this is all the explanation that I have already done. All right, look at this picture and read out this one. Don't go to the results, don't go to the interpretation, because we have to talk about the interpretation and results on our own. So it be
So we
just
can edit
read
now.
this. Okay, what is the coleoptile? Let me hold on. The coleoptile over here is basically the white seed of the wheat seed, um, sh- the shoot of wheat that is covered by the sheath, and that sheath is called as the coleoptile. Okay, and from the coleoptile, the auxin is being released here. Auxin is being released here from the coleoptile. So now read it.
Wheat and other grass species belong to the monocotyledon group of flowering plants. When wheat seeds germinate, start to grow, they produce a shoot covered by a protective sheath called coleoptile. This helps prevent damage to the new leaves as they push through soil. The coleoptile shows responses to light and gravity in a similar way to other plant forms. Wheat coleoptiles only take 2 or 3 days to grow and they show responses very quickly, so they are ideal for focusing experiments. The tip of the coleoptile, where it is expected that auxins would be produced, can be cut off without killing the plant, effectively removing the source of the auxin.
Okay. Now look at this particular setup very closely. This one. Very closely.
Okay,
All right. Now what do you think would happen in A?
so it would not grow because it doesn't have a tip in it.
It would not grow because
There's no tip.
there is no
Tip,
there is no coleoptile, or there is no tip. Okay. And obviously when it would not grow, what is the cause of, like, if the tip is not there, why it would not grow? Remember, you have removed the source of oxygen.
yeah, so it won't.
You have, yes, the growth was dependent upon the oxygen. You have removed the source, you have removed the coleoptile, leading to the stop in the growth. No more bending, no more growth, no more gain in length. Okay, what do you think would happen to B?
It would grow as normal. because it has the oxygen. But wait, I
Hmm.
think maybe it would like it won't go straight because light is only coming from one side,
Very good. Well done. Well done. Going great. Yes. I-
leaning towards the left side.
Well then, it would grow. You did not remove the source of oxygen. You did not remove the coleoptile, which was the source of oxygen. It is supposed to be going straight, but since the light is coming from one side, it would bend towards the light. Well then, what is going to happen to C?
It will not because it wait, it would grow, but
Mhm.
like it would have the oxygen, but it won't have the sunlight. So
It would have the oxygen, but
it won't have any light because the black paper just is creating a shady area for the oxygen. The oxygen will be produced, but there's no sunlight because it's covered.
so what do you think would happen to the shape of the shoot? Should it bend or should it go straight?
It goes through,
Yeah, exactly.
but it would be very.
What is Yeah, exactly. All right. What is going to happen to D?
What do you think agar block transferred to polyethylene?
Let me tell you a bit about the agar block. Agar is a jelly.
Yeah,
Agar is a jelly-like thing, and the substances, if something is coming from above, the substances can diffuse into it, and they can also be diffused out of it. So shortly, this was the explanation. What do you think would happen here now? 6 hours later, you are transferring the agar block to the uh, back to the tip. What do you think should happen here?
I don't understand what they're doing.
You did not? Sorry?
I don't understand what they're doing in this
See,
class.
basically they are telling you that it is the coleoptile that is the source of oxygen, no other part of the plant. How is it being proved? You removed the coleoptile, you put it over the agar block, then you took off the coleoptile, took the agar block. Instead of coleoptile, you place the agar block over the D. Now, if it grows and bends to the light and bends towards light, it means that agar block had oxygen.
Mm.
It means agar block had oxygen. Since it had oxygen, where did it come from? Where did the oxygen come from uh, in the coleoptile, in the agar?
Maybe it is already present.
I'm sorry, no, it was not already present. Look closely what they did initially.
Okay, so because it's now inside the agar block.
Yes, it is now inside the agar block because initially you put the tip over the agar block. The oxygen got diffused into the agar block. Now, instead of coleoptile, you are placing the agar block over the shoot D. If it is growing, it means the actual source of oxygen is the coleoptile. Why? Because the coleoptile released the oxygen, got accumulated into the agar block. Now the agar block has become the source of oxygen, but it is not the original source. The original source was coleoptile.
Yeah.
It basically is proving that the reason of auxin production is the coleoptile.
Okay.
A, a way to prove a thing is to check it by removing. When you remove something, check its effect and try to bring that thing in the other way around, then it would prove that what is the importance of that thing. The coleoptile was first removed, then we accumulated all the auxin into the agar. The agar was replaced, and now the plant would grow and it would bend toward the light. I guess it is clear now.
Yes.
So these interpretations all have been explained by us. So it is just needed to be read. There is nothing special in this text. Got it?
Yeah.